Right, we're now even closer to having our PCR machine full of all the ingredients we need. If you recall, we had two samples, didn't we? We had one from which were from my saliva and one from cheek cells.
Yeah.
And we've just labelled up our PCR tubes. And if I remember rightly, the saliva is going to go into that one.
Yeah.
And obviously the cheek cell material will go into the other. And here we go again. There's another reason for students being equipped into the use of the micropipettes, so that that investment in time early on is going to pay dividend yet again, we hope.
Okay. So we're going to measure 20 microlitres of our cheek cells or our saliva cells in DNA into each PCR tube. It's important to keep the PCR tubes shut until you are just ready to put your sample in.
Because any contamination, this is the vital point, isn't it?
Any contamination might be amplified by other people's DNA. It's also important to wear gloves so that you don't contaminate the sample with your own DNA. Now, just going to put that 20 microlitres in there. This sample first of all.
And if you remember from before, we talked about good practice of keeping the lid closed from all the other pipette tips.
Now down to the soft stop, in with your tip. And release.
Okay.
And then into the PCR tube.
So again, you need to be fairly dexterous when you're using one hand. So that's the DNA ready for that part. And then to repeat again the DNA taken from the other. I think that's taken as read, isn't it? It'll be the same process for the other sample, which we'll do again off camera. Okay?